Hi, my name is Taylor Bright. Today I'll be talking about type 2 diabetes, which is characterized by insulin resistance of the tissues which normally take up glucose. When these tissues become insulin resistant, that means that they're no longer taking up glucose from the blood, which leads to hyperglycemia and inflammation in other tissues. This is typically this can be a result of the adipose tissue becoming too large too fast. In this case, the inflammation response which ensues is based on signaling molecules, otherwise known as cytokines. Cytokines shown here in this image over here are the pro-inflammatory ones, here are the anti-inflammatory ones. It is the goal of this study to use the reactive oxidative species typically present in these systems and generate them with cold atmospheric plasma. This is a fourth state of matter which uses ionized argon, which produces the species. The electroporation of the membrane opens up the pores by electricity and causes the reactive oxidative species to enter it, signaling the release of these molecules. When they're released from these monocytes, which are the directors of immune responses, it is our proposed reaction that these cytokines are decreased and these cytokines are increased, leading to the wound healing response rather than the inflammatory response in these cells. The methods that we use a cell line of THP1, which are human leukocytes, cold atmospheric plasma, electroporation, bioinformatic analysis using GROMACS and AutoDoc-Vina to establish the binding free energy to understand if the cytokines were going to be upregulated or downregulated. We also analyze the pH, intracellular and extracellularly, and also lattice light sheet microscopy and inflammatory array to establish the concentration of the reactive oxidative species had on the presence of the proteins. The results that we gained from it based on our bioinformatic analysis was that our temperature and reactive oxidative species load had a significant impact on the expression of the proteins. Our intracellular pH showed that the cold atmospheric plasma at 60 seconds and a high treatment of electroporation resulted in a basic pH. Our extracellular pH of the analysis of the media came to a similar result. Our lattice light sheet microscopy can be shown here and indicated that no adverse effects could be seen on fetal liver macrophages. Our inflammatory array showed IL-13, which is a typically anti-inflammatory cytokine, monocyte chemoattractant protein 1, which adds more monocytes and immune cells to the wound area, and interferon gamma, typically an inflammatory cell, all increased with the treatment of cold atmospheric plasma. Based on these conclusions, we believe that the regulation of the cold atmospheric treatment can lead to an anti-inflammatory response triggered by these signaling pathways. The lattice light sheet also demonstrated viable cells after the CAP treatment, which is contrary to literature in which cold atmospheric plasma can be used to sterilize or kill cancer cells. Future work will be based on cold culturing with beta cells, typical in type 2 diabetes studies. Thank you.